Med students have to learn about a vast amount of drugs in medical school and are expected to know about them. You might be surprised how many medicines were actually derived from nature. Many know that aspirin is derived from willow bark, but few realize just how many other medications are derived from plants. A number of very common and useful medications employed today have extremely interesting histories and were taken from nature. We show you 10 plants that led to useful and life-saving drugs, and hope you find the origins of these medications to be as fascinating as we do. Number 10. Cannabis Sativa and Dronabinol. The Cannabis Sativa plant has been part of the recent controversy regarding the legality of marijuana. While marijuana is most commonly associated with the cannabis plant, there is another extremely useful pharmaceutical that has come from it. Many know the symptoms of marijuana intoxication, including red eyes, dilated pupils, dry mouth, increased appetite, slowed reaction time, euphoria, dizziness, shallow breathing, and increased heart rate. While some of these symptoms seem unappealing, the medical community has found others to be useful in treating certain populations of patients. The drug dronabinol has been created as a synthetic form of THC to utilize some of marijuana's side effects. There are various uses for the drug, but most commonly, it is used as an appetite stimulant for patients with AIDS and as an antiemetic in patients receiving chemotherapy. While there has been some controversy regarding the use of dronabinol, it has been shown to be minimally harmful with a low potential for abuse. Who knew that giving someone the munchies could be so beneficial? Number 9. Podophyllum peltatum and Atopa. The Native Americans have recorded using the plant Podophyllum peltatum as a purgative, antiparasitic, and cathartic hundreds of years before its usefulness was officially recognized. Interestingly, the Penobscot people of Maine even appeared to be using it to treat cancer. The Iroquois additionally used it to treat snake bites and as a suicide agent. Despite this, the medical use for P. peltatum was not official in the United States until 1820, and not until 1861 in Europe. Hartmann Stalin was a Swiss pharmacologist who had made large contributions to the cancer therapy field. He had a particular proclivity for biomedical sciences, and was recruited to lead the pharmacology department in Basel in hopes of researching cancer and immunology in 1955. Once in Basel, he led the discovery of various anti-tumor agents from P. peltatum, also known as mayapple. Initially considered by chemists to be dirt, Stalin noticed that a particular extract from the podophyllum plant had interesting properties. After purifying this compound, it was found to be a new class of anti-tumor medication. Named Atopa, the medication works by stopping the tumor cell's ability to divide. It blocks a specific enzyme that cells need in order to replicate. Therefore, rapidly dividing cells such as cancer cells are heavily affected. Currently, Atopa is used to treat various cancers, especially that of the lung, and can be thanked for saving many lives. Number 8. The Calabar Bean and Physostigmine. The Efik people from the Akwa Ibom state, or modern-day southeast Nigeria, were the first to be in contact with physostigmine from the Calabar bean. Use of the Calabar bean was very common in Efik culture, as an ordeal poison for those accused of witchcraft. The milky extract of the bean was given to the accused, and if they died, the accusation of witchcraft was confirmed. If they lived, usually due to vomiting the poison out, they were declared innocent and set free. Missionaries wrote about the Efik's use of the Calabar bean, and some of the beans found their way back to Scotland. In 1855, a toxicologist named Robert Christison decided to test the poison's toxicity by consuming a bean and surviving to document what he experienced. It was studied throughout the 1860s, most notably by Douglas Argyle Robertson, who was the first to use the Calabar bean extracts medically and recorded its effects on the pupil. The most potent component from the Calabar bean was finally isolated and named physostigmine by Thomas Fraser. In 1867, Ludwig Lacour tested the extract on himself and used it to successfully treat his glaucoma. By the 1920s, Otto Loewy discovered the neurotransmitter acetylcholine and found that the Calabar bean extract worked by increasing that neurotransmitter, having profound effects on the parasympathetic nervous system. Medically, physostigmine does increase the amount of the neurotransmitter acetylcholine by blocking the enzyme acetylcholinesterase, which breaks it down. It is especially useful in treating the disease myasthenia gravis and has been more recently used to treat Alzheimer's, as it has the ability to cross the blood-brain barrier. Number 7. Meadow saffron and colchicine. The use of the plant Colchicum autumnale or meadow saffron for medical problems has been recorded as far back as 1500 BC on the ancient Egyptian Ebers papyrus for rheumatism and swelling. Since then, C. autumnale has been a treatment for other maladies such as gout, familial Mediterranean fever, Behçet's disease, and pericarditis. It works in a similar way to Taxol, as it blocks microtubules. As early as the 1st century AD, Colchicum autumnale was being described as a treatment for gout by Pedanius Dioscorides. Gout is a type of arthritis characterized by needle-shaped crystals building up in the joints, causing sudden pain attacks, swelling, and redness. Others, such as Alexander of Tralles, Persian physician Avicenna, and Ambroise Paré have also recommended Colchicum autumnale as a treatment for gout. Colchicine itself was isolated from Colchicum autumnale in 1820 by French chemists P.S. Peltier and J.B. Caventou. It was later purified by P.L. Geiger in 1833. Despite its long history of being effective, colchicine actually had no FDA-approved prescribing information, dosage recommendation, or drug interaction warnings until as recently as 2009. Number 6. Indian snakeroot and reserpine. Rowolfia serpentina is a plant that was known in India for its medicinal purposes long before its discovery by the Western world. Georg Rumpf, a botanist with the Dutch East India Trading Company, first noticed the plant in 1755 during his travels. He recorded it as being used as a treatment for insanity in South Asia. Extracts from the roots of the Indian snakeroot were sold cheaply in markets all over India as Pagalon ki dawa or drugs for the mad. In addition, it was also used by mothers in eastern India to put their crying babies to sleep, as well as a treatment for labor, snake bites, fever, and intestinal problems. Mahatma Gandhi reportedly used extracts from the roots as a tranquilizer as well. By the early 20th century, India undertook efforts to standardize and research the pharmacologic properties of sarpaganda. Professor Salimuzzaman Siddiq began systematic research on the active constituents of the roots and root bark in 1927. Dr. Kartik Chandra Bose and Gananath Sen, two leading physicians from Calcutta, also independently noted the use of the extract to treat high blood pressure and insanity. Dr. Rustom Vakil, known as the father of modern cardiology in India, popularized the use of the plant to treat high blood pressure. Isolated in 1952 from the dried root of R. serpentina, reserpine quickly became popular in Western medicine. It became the first drug ever to successfully show antidepressant properties in a randomized placebo-controlled trial. Though it is rarely used today due to its immense side effect profile, it was critical in furthering our understanding of the role of neurotransmitters in depression and blood pressure. Number 5. Indian hemp and pilocarpine. As settlers began coming to the New World in the early 1600s, they noticed that the indigenous tribes of Brazil had a vast knowledge of the medicinal uses of local plants. One plant in particular, Pilocarpus jabarandi, was used to treat a variety of maladies but most commonly for fever. It was found that the leaves could trigger profuse sweating, salivation, and urination as a way to rid the body of toxins. The name jabarandi even comes from the Tupi translation for what causes slobbering. In the 1870s, Pilocarpus jabarandi was incorporated into Western medicine and became a popular treatment for intestinal problems, lung problems, fever, skin issues, kidney disease, and edema in Europe. Surprisingly, the plant was also found to be an effective antidote to deadly nightshade poisoning. By 1875, pilocarpine was isolated from the plant and found to be the main culprit behind its effects. This was discovered almost simultaneously by two different researchers, one in France and one in England. Pilocarpine was soon found to be an extremely effective treatment for glaucoma by decreasing the pressure in the eye. Even today, it remains a very popular and widely used treatment for glaucoma, as well as a means to induce perspiration when trying to diagnose cystic fibrosis. Laboratories still haven't been able to fully replicate and synthesize the pilocarpine found in Pilocarpus jabarandi. This plant remains one of Brazil's largest and most important exports. Number 4. the Pacific yew tree and Paclitaxel. Researchers are continuously searching for new and innovative ways to fight cancer. Sometimes the treatments that they are searching for may be much closer to home than they realize. In 1955, the National Cancer Institute created the Cancer Chemotherapy National Service Center in hopes of finding new cancer treatments. In the 1960s, the Cancer Chemotherapy National Service Center looked to partner with the U.S. Department of Agriculture to search for these cures within nature. Over the course of about 20 years, 30,000 natural plant and animal products were tested. Out of the 30,000 samples, one was found to be pivotal in the treatment of cancer. Two researchers, Dr. Monroe Wall and Mansukh Wani, discovered that the extracts from the bark of the Pacific yew tree, indigenous to the Pacific Northwest, were toxic to tumor cells. Later, it was found that the toxic compound is actually synthesized by a fungus within the bark. Thus, the new chemotherapy drug known as Paclitaxel was born. Paclitaxel, brand name Taxol, is commonly to treat breast and ovarian cancer. Medically, it works by blocking microtubules, which basically stops the cancer cells from being able to divide and grow. Since its discovery, Paclitaxel has become a big part of cancer treatment and saved millions of lives. Number 3. Deadly Nightshade and Atropine. Atropa belladonna, commonly known as belladonna or deadly nightshade, is an herb that has been used for many centuries by many people to treat a wide variety of maladies. The plant is native to Europe, North Africa, and West Asia but has been more recently introduced to Canada and the United States. Prior to the Middle Ages, the herb was used as an anesthetic for surgery. Its deadly toxicity enabled its use as a poison for political enemies or on the tip of an arrow by the military in ancient Rome. During the Middle Ages, the deadly nightshade plant became very popular for cosmetic purposes. Venetian women would use it to redden the pigment of their skin as a type of blush. Another common use for the herb was for dilating the pupils of women in order to make them more seductive and attractive. The herb obtained the name belladonna, meaning beautiful lady, exactly because of this use. Despite these more benign functions, many quickly became aware of the herb's more deadly abilities. It was later utilized by assassins and criminals, as well as witches to make poison. Despite years of its use as a poison and cosmetic, it was soon realized that belladonna had more of an ability to help than previously realized. It could be used as a pain reliever, muscle relaxant, anti-inflammatory, whooping cough treatment, and hay fever treatment. In the 1930s, the therapeutic component of belladonna, known as atropine, was isolated. Belladonna by itself does not have approved medical uses, but atropine has since become an extremely useful medication in the medical community. Atropine is known as an anticholinergic, meaning it blocks the effects of the neurotransmitter acetylcholine. Its mechanism of action is basically opposite to that of physostigmine. Because of this, atropine can cause pupil dilation, increased heart rate, and decreased secretions. In addition to its uses of raising heart rate and decreasing saliva prior to surgery, it can also be used to reverse certain overdoses. Various derivatives of atropine have also been developed for other medical uses. For example, tiotropium and ipratropium bromide are used in various lung disorders. Number 2. The cinchona tree and quinine. Found in the bark of the cinchona tree in South America, quinine was initially used by the Quechua as a muscle relaxant. It was then brought to Europe by the Jesuits, and by 1570, the Spanish had become aware of the cinchona bark's medicinal properties. Nicolas Menards and Juan Fregoso recorded that it could be used as a treatment for diarrhea. Despite the varied ancient uses for quinine, the big discovery for its use came in the early 17th century. The marshes and swamps surrounding Rome in the early 17th century were teeming with malaria-ridden mosquitoes. Malaria is a mosquito-borne infection caused by parasitic protozoans. Symptoms include fever, fatigue, vomiting, headache, jaundice, seizures, and eventually death. Malaria led to the deaths of many popes, cardinals, and citizens at the time. Agostino Salambrino, a Jesuit apothecary, had seen the cinchona bark being used for the shivering phase of malaria. At the time, Salambrino did not know that the bark's effect on malaria was unrelated to its effect on rigors, but regardless, he brought it to Rome. Over the years, cinchona bark became one of the most valuable exports from Peru, even curing King Charles II. In 1737, Charles Marie de la Condamine discovered the most potent component of cinchona bark, and it was later isolated by Pierre-Joseph Peltier and Joseph Caventou in 1820. The extract was named quinine, based on the Incan word quina, meaning bark or holy bark. Large-scale malaria prophylaxis with quinine began around 1850. The drug actually played a very significant role in African colonization by Europeans. In the early 19th century, Peru tried to outlaw the export of cinchona bark, seeds, and saplings to maintain their monopoly. Fortunately for the world, the Dutch were successful at growing the tree in their Indonesian plantations, and soon became the main supplier. During World War II, the Allies were cut off from quinine when Germany conquered the Netherlands, and Japan controlled Indonesia and the Philippines. The United States was eventually able to obtain 4 million seeds from the Philippines, but not before thousands of Allied troops died from malaria in Africa and the South Pacific. Thousands of Japanese troops also died despite their control due to ineffective manufacturing of quinine. Since its discovery, quinine has played a role in saving millions of lives, as well as having major effects on wars, colonization, and history in general. It has since been replaced as the first-line treatment for malaria by newer drugs, in 2006 by the World Health Organization. Quinine can also be used for other diseases, such as babesiosis, restless leg syndrome, lupus and arthritis. Number 1. Foxglove and Digoxin. Digoxin was once a mainstay treatment for heart failure and arrhythmia. It works by slowing the patient's heart rate but increasing the heart's contraction intensity. Unfortunately, the drug has a very narrow therapeutic index, meaning that it can be extremely easy to overdose, with disastrous effects. Digoxin's discovery by Scottish Dr. William Withering occurred in 1775. He was working as a physician when a patient came to him suffering from a bad heart condition. Withering had nothing to offer the man, as there was no acceptable treatment for heart failure at the time. Thinking he was going to die, the patient went to a town gypsy and miraculously improved after being given an herbal remedy. After seeing this, Dr. Withering searched for the gypsy, eventually finding her and demanding to know what was in her remedy. After Dr. Withering bargained with the gypsy, she finally revealed many things within the remedy, but Digitalis purpurea, or foxglove, was the main ingredient. The potency of foxglove was already well known, as it had been used as a poison in medieval trials by ordeal, as well as externally applied to heal wounds. Withering immediately went to work testing variations of the foxglove extract on 163 patients. He eventually found that dried, powdered leaves gave him the most successful results, and it was first officially used in 1785. Even though it is not as commonly used now, digoxin was revolutionary in its ability to help those with heart failure.